Welcome, everybody, to our monthly Science of Alt Protein seminar. It is a joy to have you here with us. For those of you who are new to GFI, the Good Food Institute is a 501c3 nonprofit organization. We are 100% driven by philanthropy, and our singular goal is to accelerate the alternative protein field as quickly as possible so that we can build a sustainable, secure, and just protein supply. And we work across many different pillars of work: science and technology, corporate engagement, policy, all to make sure that folks across the continuum, from the smallest entrepreneurs to the biggest universities and corporations and governments in the world, are able to have the resources that they need to advance alternative proteins. And my name is Amy Huang. I am the Associate Director of Scientific Ecosystems at the Good Food Institute. So with that, I will introduce our lovely speaker today. We are joined by Dr. Filiz Koksel, an assistant professor at the Department of Food and Human Nutritional Sciences at the University of Manitoba. Dr. Koksel is a food scientist with expertise in food processing and non-destructive assessments of food quality, and her research program aims to tackle issues related to this ever-increasing demand for sustainable, high-quality plant-based foods. Dr. Koksel's creativity in exploring new approaches in these areas is particularly noteworthy because her program is at the interface between food processing and material science, which is a critically underserved discipline in the alternative protein discourse. So to bring more Canadian crops from the field to our tables, Dr. Koksel's program looks at several operations like milling, mixing, sheeting, baking, texturization, and extrusion to transform these crops into foods that taste great and have really strong nutritional value. And so in today's talk, Dr. Koksel will be diving into her GFI-funded research to obtain real-time ultrasonic information on extruded plant-based meats and link this information to the physical and nutritional quality of the end product. Lots for our community to learn from her today, and delighted to be joined by her. So with that, I will pass it over to Filiz to get started. Thank you so much for being with us here today.
Thank you very much, Amy. It was a great introduction. I'd like to start with thanking the Good Food Institute for supporting this research project. It's really helped me to, it really shaped a part of my research program at the University of Manitoba. I used to do lots of work on extrusion, but mostly on the puff snacks and breakfast cereal sides of things, maybe with added fibers and proteins. And I already had an interest in protein-enriched plant-based foods. But with this project that GFI funded, my focus kind of shifted from breakfast cereals and puff snacks to more on the plant-based meat alternative side. It did shift my research program, but also at the personal level, it shifted some of my interests. And I am now involved in a startup company in South America where we're focusing on development of new plant-based meat alternatives. So with that, I'll dive into my presentation and some of them, and I think given the audience, the concerns related to the food security and sustainability issues and the growing world population and the ever-increasing need for nutritionally dense foods, which also have lower water and carbon footprints, is something that we've been all talking about for a while now. And keeping these in mind, one of the emerging food markets that's experiencing a definite clear growth trend is plant-based meats. And plant-based meats category has several different products in it, including vegan or vegetarian patties, sausages, fox meats, chunks, etc. And while there are several different methods to make produce plant-based meats, some of them being more traditional like tofu and tempeh, and also other techniques being used such as shear cell, etc. But the most common technique that's used for producing fibrous texturized plant-based meats is extrusion cooking. And I'm assuming most of you in this talk are familiar with the process, but for those of you who aren't, extrusion cooking of plant-based meats start with a dry feed that generally has a high protein content. So it could be a protein concentrate, a protein isolate, or a mix of these with some other materials. And then the dry materials are fed into an extruder, and then some liquids, mainly water, is added to the barrel. And in the extruder barrel, these materials are blended and pushed towards from the feeder end to the die end. In this slide, you can see a twin screw extruder that's commonly used for plant-based meat extrusion. And as the material mix is being pushed from one end to the other, you're also applying heat, you're applying shear, and you're transforming some of the macromolecules in your raw materials like the starch and the proteins. And the material then reaches a long cooling die for the production of high moisture methanol logs, which is the main topic of today's talk. And then as the material is being pushed, more changes in the proteins occur, which is responsible from this fibrous structure and chewy, juicy texture of high moisture methanol logs. So for the food scientists and the food industry, the trick is to manipulate extrusion conditions using a plant protein rich raw material to obtain a product that is able to mimic the fiber structure, chewy and juicy texture of meat. However, the extrusion process itself is not a very easy process. Generally, it's been considered by many researchers in the field as a black box, because you start with a feed, you end up with an end product, and while doing so, you have the ability as a processor to change some of the processing conditions, like the screw speed of the extruder, or the dry feed rate, the wet feed rate, the water injection rate, the temperature profile across the barrel and across the die. And all of these process conditions that the processor can manipulate also indirectly affect some other things. For example, how much pressure is going to build up as the material is being pushed from one end towards the die end. And with the changes in screw configuration or screw speed, then you're also affecting the shear forces inside the barrel, and also with the die dimensions, the shape and the dimensions of the die that you use. Again, you're able to manipulate that shear forces, this time within the die. And all of these interact all together with the processing conditions to produce the end product that you're trying to make. And in addition to all of these complex interactions, producing one product in one lab, for example, in my case at the University of Manitoba, using exactly the same conditions does not necessarily mean that somewhere else in, let's say, British Columbia is going to be able to make exactly the same product, because the end product characteristics can also depend on the extruder capacity and and scale, if you can scale up from benchtop to pilot to industrial scales of extrusion. On top of these, you can use several different ingredients. While the most common ingredients for the plant-based, high moisture plant-based meat analogs, we use like soy and maybe gluten, peas. There's also other proteins that have been emerging recently, and every time you change your formula, or if you blend in a new formula, be it lentil, pumpkin, faba bean, potato, barley protein, all of these affect the material properties, for example, the viscosity of the melt inside that barrel. And on top of that, depending on how you manipulate the raw material properties along with processing conditions and your extruder, you can produce anything from a high moisture meat analog or a lower moisture meat analog, also known as texturized vegetable proteins, and protein enriched puff snacks. So with the high moisture meat analogs, generally, these types of products can fully replace animal proteins with texturized vegetable proteins, or TVPs, or low moisture meat analogs. Usually these products are drier and they need rehydration before being incorporated into foods. And I would say most commonly they are used for partially replacing animal meat, but there's also instances that where you can see they're used to fully replace animal meat, for example, in taco fillings. In today's talk, just to keep things a little simpler, I will focus on one type of raw material mix. So I will not be looking at compositional changes in my raw material mix. I'm talking about some experiments we performed on a mix that included soy protein and wheat gluten. And on this side, I will be focusing on the production of high moisture meat analogs, and the discussion of the techniques that I will talk about today can be expanded to include texturized vegetable proteins, the lower moisture meat alternatives. But for the sake of simplicity, today's talk is just on high moisture meat analogs, or HMMAs. And for those of you who are in the industry for production of high moisture meat analogs, or doing research on this topic, you'll very easily, you'll be able to tell me one of the biggest issues of producing HMMAs is achieving a texture, a microstructure of these plant-based meat alternatives, similar to your chicken breast, beef, fish, or other seafood products. Assessment of quality for texture and microstructure keeps coming up in the literature. We're always looking for ways to assess the quality and making sure that the products, the high moisture meat analog products that we're extruding, are similar to one of these animal-based meat products. But with more advances in the field, we also see that there's also efforts to make sure that the color and the nutritional quality of these plant-based meat alternatives matches or exceeds the expectations when compared to animal-based meats. Oops. And on top of this, one of the issues that the food industry is having is on the production side, where generally, for example, when you're measuring the texture of a plant-based meat alternative, you produce your product, you have your meat alternative, and then you cut little pieces of it, and then you take it to a texture analyzer, measure the texture offline look at your results, and then say, okay, this is not as chewy, or, it's not as soft as I want it to be. And then you go back to extrusion, and then you try to, change some of these parameters like screw speed or temperature profile, as I mentioned. Maybe you add in, different ingredients to help you get to that texture, and then try again. And then once you get a new product, you again stop extrusion, you cut a little piece, you go and test it on an instrument, or you do sensory analysis. And that really takes a lot from processing time, because you need to do things offline. And another issue in the food industry is that when you're testing that meat analog, you take a piece, so you're touching that meat analog, and whatever you're testing is going to go to garbage. Because there's human involvement and carrying the food from one place to another, so it's not sanitary. So one of the main focuses of my talk today is related to addressing these two concerns for the food industry, where I'll introduce a method where we do not have to worry about sanitary conditions or downtime during processing. And for this, today I'm introducing an online ultrasonic system that can be mounted on an extruder or placed at the die exit. So here you can see the one that says contact transducers. This is mounted on the extruder's long cooling die. So this will be the mounted option. Alternatively, you can place non-contact air-coupled transducers close to the die exit to characterize the properties of your meat alternative. And this method is also able to provide real-time information on how different food processing parameters affect the final product quality. So here the processing parameters, could be anything from dry feed rate, water injection rate, barrel temperature profile, die temperature profile, screw configuration, or screw speed. And the ultrasonic system can also provide real-time information on food's mechanical properties without any contact with the food product, for sanitary reasons, as I explained. And in both of these options, either the contact or the non-contact, you're not really in touch with the food material itself. Even with the contact transducers, the contact is between the long cooling die and the ultrasonic transducers, and not with the food. And here with the air coupled transducers, the transducers are not touching the food material, so the plant-based meat alternative is just flowing in between the two ultrasonic transducers. And finally, this online ultrasonic system, hopefully one day, will ensure end product consistency and prevent loss of materials and downtime periods in the food industry. So the technique, the use of low intensity ultrasonics to measure the properties of any food is not something that I discovered yesterday. This tool has been utilized in the food industry for a while now, and our team in particular has been involved in some of that research. And on this slide, you can see two examples. One of them is characterizing the bubbles in bread dough and how they evolve as a function of time during baking. And the second one is characterizing the properties of noodle dough during production. And both of these food materials, as you know, are optically opaque. So ultrasound gives you the advantage of being able to work with foods that are not transparent to light. So here on this figure, you can see a noodle sheet flowing during noodle manufacturing process, and you can see two ultrasonic transducers, one higher up here and one down here. And on this bottom figure here, again, there's like a sheet, and then you have two transducers, and these are the exact same transducers we use for the work that we do on extrusion cooking of plant-based meat alternatives. And the outcomes of these, these initial studies was that using low intensity ultrasonics, we were able to detect changes in product properties, changes in bread and noodle properties as a function of ingredients. For example, if you increase or decrease salt content, if you increase or decrease water content, you can get that information extracted from the changes in the ultrasonic signal. And also for the noodle study, we were able to see that ultrasound was able to pick up differences in the thickness of noodles, which is a very important parameter for the noodle making industry. The advantage was that instead of measuring the thickness using like a caliper, cutting a piece, we can do it in a non-contact way. And you can really see here that nothing is touching the material that's flowing in between the transducers. So let's start with what ultrasound is. Basically, ultrasound waves are mechanical pressure waves with frequencies higher than the upper audible limit of human hearing. And it's very commonly used. For example, imaging in medicine or non-destructive testing of structures, seeing if there's any cracks in pipelines as an example, or even for locating prey by animals such as bats. And the application of low intensity ultrasonics has the advantages that the technique is non-destructive, it's really fast, it's relatively non-expensive compared to some of the other techniques that can be used to determine the mechanical properties of food materials. And very importantly, ultrasonics, low intensity ultrasonics can provide some answers for optically opaque materials. And since many food materials are optically opaque, this is a great advantage. Here, I'd like to take a second to emphasize that the ultrasonics that we use are low intensity, so they're non-destructive. So they're not supposed to cause any changes in the structure or the composition of the material being tested. Imagine it like this. So when if you're pregnant, if you're getting an ultrasound image of the baby, so similarly, that ultrasound has no effect on the baby, right? So, or like when you're testing pipelines, you just test the cracks and you're not really changing what's flowing inside that pipe. So it's the same idea. And I think in the food industry, sometimes when you say ultrasound, since higher intensity ultrasound is also commonly used for making changes in, for example, protein structure, or even cleaning some of the food equipment. So that's really different, and it's at the higher intensity end of the spectrum. And what I'm talking here today is low intensity and non-destructive. So with that, here on this slide, you can see that schematic of the extruder again, and we're really focusing on this area where everything is air coupled. So today's talk is not on the contact transducers, but only on the air coupled transducers. So if you magnify it here, the extruded or the plant-based methanol that's flowing, that's leaving the long cooling die, is the one with the dark orange brownish color. And then we have one transducer here that's emitting the ultrasonic wave. And then there's another transducer on the other side of the methanol that's receiving the signal. And as you can see here, I use like a blue and somewhat higher amplitude signal that's being emitted, and then it travels through your plant-based meat alternative, and then it changes its shape. The signal is changed now. It's smaller, maybe it's became a little wider, at least in this example. And then this new signal that has been transmitted through the extruded is now picked up by this receiving second transducer. And then that received signal is sent to a computer for analysis. And we're generally looking at two ultrasonic parameters to help us understand what's going on. So these two parameters are velocity and attenuation. So velocity is easier to relate to. It's how fast the sound travels in the plant-based meat. And attenuation is how much of that sound is lost in the plant-based meat. It could be due to scattering, it could be due to absorption. And the idea here is to, at the end, find some correlations between the texturization behavior of the plant-based meat alternative and the ultrasonic properties. And how that happens is that if we again just magnify onto the extrudate, so now this becomes the extrudate here, and imagine it has a thickness D. So this signal is coming from the emitting transducer. The emitting transducer is somewhere here. So it comes, it travels through air, and then it enters the extrudate. It can travel the thickness of the extrudate. It can leave the extrudate, and the transmitted signal can be picked up by the receiving transducer and sent to a computer to be analyzed. But alternatively, the same signal can travel once, be reflected from the interface of the extrudate and air. So this is the interface. So come this way and then just leave on this side. Or it can be reflected once again from this surface, making 3D, so three times the thickness of the extrudate, 1, 2, 3, and then be received by the second transducer. Or it could just keep going 1, 2, 3, 4, 5, and then be received by the second transducer. And it goes like 1 thickness, 3 times the thickness, 5 times the thickness, 7 times the thickness. And this, I'll come back to this traveling distances, because it's going to be important when we're analyzing our signal. So just to take a step back and review the ultrasonic parameters, because we'll keep hearing these. The attenuation coefficient is how much of that sound signal that's lost due to scattering or absorption. So if there's some scatterers, for example, bubbles in your matrix, or the matrix itself absorbs the sound waves, then you'll have a higher attenuation. And for ultrasonic velocity, if the sound waves travel faster in your material of interest, then you'll have a high ultrasonic velocity, or the opposite if the waves are traveling slowly. And both of these ultrasonic parameters depend on the material through which the sound waves is propagating. So the mechanical properties of the meat analog, and if there are scatterers like bubbles, that's also very important, and the density of the meat analog. And this density can very simply be measured independently, and I'll talk about that too. So just to give you a magnified version of where we're testing the ultrasonic properties. So just over here is the dye end, and our meat analog is flowing. So this is a picture from our lab, and you can see one of the transducers, so the emitting transducers. You cannot see the bottom one here, but in this schematic, you can see that here is the emitting transducer, here is the ultrasonic signal that's passing through the meat analog, and then the second transducer here is picking this up. In an industrial setting, you can simply use a conveyor belt to keep the material flowing. But in our case, we didn't have space, so we just used this inclined plane. We also made sure that we're only measuring the signal that's passing through the meat alternative by damping the signals using an inclined aluminum plate at the two sides of the meat analog. So as I already mentioned, we performed the ultrasonic measurements in transmission mode. So we're getting the information from the signal that has been transmitted through the meat analog. And there are other ways to do it, for example, in the reflection mode, but we're only focusing on the transmission mode results today. And we use two transducers with active surface areas of less than 40 millimeters each. And just to give you an idea, our plant-based meat analogs were around 50 millimeters wide. So the active surface of the transducers are slightly, like the diameter of the transducers are slightly less compared to the width of the plant-based meat alternatives. And we used a frequency range of 250 to 450 kilohertz with a central frequency of approximately 400 kilohertz. So the details of signal acquisition and recording and analysis hopefully will be out in a couple months in an open access publication we submitted, and be sure to check that out on the GFI website when it's ready to be published. But all this information will be available to you if you're interested. So in our laboratory setting, we have a small lab scale twin screw extruder. Here is our feed hopper, the motor, here is our barrel, and here is our long cooling die. And the material is being pushed from here. So the dry material blended with water in the barrel and then pushed through a long cooling die. And then here at the die end, we have like a slit opening. And here is where the extruded or the plant-based meat alternative is going to be flowing from. And if we were to just magnify here on this next picture, you can see the end of that long cooling die. And here is our plant-based meat alternative. And here are the two transducers. One emitting the ultrasonic signal. The signal travels through the meat alternative, and then it will be picked up by a second transducer here. So this is the non-contact setup. And if we were using a setup that's mounted on the extruder, that would have been here on the long cooling die, which will be a topic for another presentation maybe. So what do we, how do we proceed with the ultrasonic analysis? We collect ultrasonic signal just through air. So we have a emitting transducer. Here's our signal, and then receive the sound signal just going through air. So that's our reference. And in this one, we have the plant-based meat alternative. Again, the emitting and receiving transducers, same signal, but this time signal changes as it propagates and as it's transmitted through the plant-based meat alternative, and then it's received here. So on this figure, you have time and the signal amplitude for the signal that passed through the meat alternative. And on this other y-axis, you have the reference signal. So here, the red one here is the reference, and the blue signal is through the meat alternative. So in our experiment, we found that the signal through the meat alternative had very low attenuation. So there weren't many scatterers, or the signal was not absorbed much by the meat analog itself. So we saw that there were multiple reflections. And going back to this figure again, so this is my plant-based meat alternative. So the signal arrives, and then it can travel through the meat alternative, be transmitted, and then be picked up by the emitting transducer. So it's this first peak here. So it's not the only option. The signal arrives, it travels the thickness of the meat analog, it's reflected back from the extruded air interface here, and then when it reaches the other end of the extruded, it's reflected back again, and then leaves the plant-based meat alternative, making a total of 3 thickness distance. And then that signal is received again as the second peak. Another alternative, the same signal can do the 5 Ds, so reflected back and forth, and then be transmitted. And then it travels 5 Ds and it will be this peak. So 1 thickness, 3 thicknesses of the extrudate, 5 thicknesses, 7, 9, and it goes like that. And as you can see, the signal amplitude goes down as it keeps traveling through the meat analog, and with that absorption and scattering, slowly the signal amplitude decays. And from that information, knowing that attenuation being very, very low, I will focus the rest of this presentation on the velocity of sound. Again, it's how fast the sound travels in the meat analog. And there are 2 ways to measure the velocity of sound in the plant-based meat analog. The first one is extracting it from the time-dependent ultrasonic signal using the peaks of the multiple reflections. So going back 1 slide, so I'm talking about this peak, this peak, this peak, this peak. So multiple peaks, multiple reflections. So we can simply calculate the velocity by dividing the twice the thickness of the meat analog divided by the average time difference between the 2 consecutive peaks, because 1D to 3D there is 2 thicknesses that the signal is traveling. The second method is obtaining the velocity by comparing the time of flight of the signal that propagated solely in air, so that's our reference, and the signal going straight through the meat analog. So this time, what we're doing is comparing this signal here, the first one received, with the reference signal, the red signal on this figure. And that calculation involves the arrival times. So how long it take for the blue one to arrive, this first peak, versus how long it took for the red one to arrive at this peak. And again, the thickness of the meat analog and velocity of air, velocity of sound waves in air. And this velocity of sound in air is something that you can look up from the literature. And at the conditions at room temperature and atmospheric pressure we used, it was 346 meters per second. So it's constant throughout all of these measurements. So here, what you do not know is the thickness of the meat analog. And if you knew thickness, you could simply calculate the velocity from either method. So how can you measure the thickness of a meat analog? So you could cut a piece and then measure the thickness using a caliper of some sort, but that's really defeating the purpose of doing things non-contact. Can we measure the thickness of the meat analog and the velocity in a non-contact way? So in principle, the 2 methods must give the same velocity. So you can use either method to determine the velocity if you know the thickness of the extrudate. But since we didn't want to just stop and measure the extrudate thickness, you could simply plot the dependence of velocity on the thickness of the extrudate. And it just becomes a system of 2 linear equations that you can solve for, and you can get the velocity of sound waves and the density that's just based on the time and velocity of sound waves in air. So these 2 equations enable direct calculation of the velocity and the extrudate thickness from the arrival times of the ultrasonic pulses and the known speed of sound in air. So what we do is we use both of these methods, and graphs group velocity versus meat analog thickness. And for example, for one extrudate that was produced at 70% feed moisture. So one method, results of one method here, results of one method here, both in red, and the intersection here gives the accurate extrudate thickness and the velocity of the sound in that meat analog sample. Similarly, for a different moisture content, you can see we use the blue curves, each curve from one of the methods. And with that, you can see that we're able to detect changes in the thickness of extrudates and changes in the velocity of sound in the meat analog that's made from, could be different ingredients. In this case, it was different moisture contents. So we're for the first time for meat analogs, we're showing a method and non-contact and sanitary alternative way of accessing the thickness of a meat analog product in real time. So I'll go back to some of the ultrasonic measurements, but I want to just give some more information about how we performed extrusion. So we use the fixed composition that contains soy protein and wheat gluten with an overall protein content approximately around 70%. And then our extruder is very small. So we're using a 0.5 kilograms per hour of dry material, but we tested quite a wide range of moisture contents, over 20%, ranging from 58 to 70%. We fixed the barrel temperature for the results here, and we fixed the long cooling die temperature. And here is what our meat analog look like. You can obviously see the fiber formation and the material that can mimic a type of meat alternative, that can mimic a type of animal-based meat. And the dimensions of our extrudates were 5 millimeters thickness by 50 millimeters width, which is the dimensions of the slit of the long cooling die. So we perform extrusion, and then during extrusion, we perform ultrasonic measurements online. And after extrusion, 24 hours later, we tested the meat analog samples using ultrasonics again. So we have data for online and 24 hours after extrusion, just to see if there's aging effects. We also measured density of the materials offline independently, because that's going to tell us compositional changes. That's going to confirm compositional changes and confirm if we have bubbles present or absent in a meat analog, because air density is much smaller than the density of rest of the ingredients. So if you have bubbles, the density will be much lower. We also measured texture profile of the meat analog. So this is a very common way to measure the textural properties of any food material. It's an offline method. So you perform the processing and then you take pieces like we have here. So we cut a little piece of a meat analog, and we're using a blade probe to cut into the meat alternative in different directions. One of them is in the longitudinal direction, which is along the extrusion flow. And the other one is the transverse direction, which is across the extrusion flow. And that usually tells you gives you an understanding of if your material is texturized or not. So the ultrasonic experiments are online or offline 24 hours after, and the texture profile analysis are offline 24 hours after extrusion. So that gives us the ability to compare textural quality 24 hours and ultrasonic parameters 24 hours. But also comparing online and 24-hour ultrasonic signals, we're also able to get a better understanding whether there were some changes related to aging. So I think this is the main results part of the project, where we're trying to link ultrasonics to texture. So we have our ultrasonic velocity on the y-axis with moisture content of the metan log on the other axis. So this result is only from ultrasonic analysis. So we have the online data we collected, which is the blue squares, and then the velocity of the metan logs 24 hours after extrusion experiments. On the second figure in the middle here, this data is collected only by texture profile analysis performed offline. So you have the peak cutting force in either direction, longitudinal or transverse, and you can see they're really similar to each other as a function of moisture content. So this is offline and only from texture. And on this third figure here, we have combined the results from ultrasonics and texture. We have the ultrasonic velocity on y-axis and the longitudinal peak cutting force on the x-axis. So the longitudinal peak cutting force is from texture, and the group velocity is from ultrasound. And this was to explore the link between the velocity and ultrasonic velocity and texture. And as you can see, our data clearly suggested a really good correlation with an R-square of 0.95. And these two parameters are positively correlated. So just to magnify here on this last figure a little bit more, the fact that the ultrasonic velocity and the longitudinal peak cutting force correlated is not a really big surprise, because there's a link between ultrasonic velocity and mechanical moduli. So ultrasonic velocity can be defined as the square root of longitudinal modulus divided by the density of the metan log. And the longitudinal modulus here is a function of the bulk modulus, which is the inverse of compressibility, plus 4/3 shear modulus. So again, going back to some of those mechanical and rheological properties of that material, the ultrasonic properties will tell us information about the mechanical properties of that material. And since we were performing a cutting action with the texture profile analysis, so the cutting involves some compression. For example, at the first time that the blade touches your meat alternative, it starts compressing, and then it enters the material, and then it'll shear a little bit. So it's a mix of compression and shear, and that is reflected in the longitudinal modulus, which goes into the formula for calculation of the ultrasonic velocity. So with that information, we're linking product texture to ultrasonic properties without the need for offline experiments. So what does that mean? We can simply measure the group velocity, and then since these are very well correlated, without the need for offline textural measurements, we'll have an idea about the textural properties of that food material. And I think both from sanitary perspective and downtime related concerns, this could also be a game changer for the food industry that uses extruders. Focusing just a little bit more on the longitudinal modulus here, which is a function of the bulk modulus and shear modulus. This can be measured directly using ultrasonic velocity if we measure density independently, which is very easy. It can very easily be done using a water displacement method. And we see that it correlates, the longitudinal modulus correlates very well with the moisture content of meat analogs. We've also done some work on nutritional quality and linking ultrasonics to nutritional quality. We looked at in vitro protein digestibility. We looked at amino acid scores. And by taking the product of the in vitro protein digestibility and the amino acid scores, we calculated the in vitro protein digestibility corrected amino acid score, in short, in vitro PDCAS. What we found was overall lysine in our in the raw material blend that we used was a limiting amino acid across all different treatments, which are all moisture content. And we saw that the lowest amino acid score was observed for the lowest feed moisture content here. So that's no surprise. And we found that when you increase the feed moisture content, the levels of amino acids increase because it becomes a less intense process. And the highest amino acid score was found at the highest moisture content. We also found that the feed moistures did not really impact the in vitro protein digestibility. And since in vitro protein digestibility corrected amino acid score is a function of amino acid score and in vitro protein digestibility, the results of the IV PDCAS just mirrored that increase with moisture, just mirrored the results that we got for amino acid score. When we look at correlations between nutritional quality and ultrasonics, our highest correlation, strongest correlation was between the longitudinal modulus that was measured online during extrusion processing and in vitro PDCAS values, but our correlations were not as strong as the ones for texture. We also found that there's an inverse relationship between ultrasonic velocity that's measured online and measures of protein quality like IVPD or IVPD-Cas. And moving forward, we're thinking of testing different feed mixtures with different compositions, different amino acid profiles to predict real-time processing effects on protein quality and detecting that with ultrasonics. So here is a slide on some of the conclusions and some take-home messages. Today, I talked about the feasibility of using a non-contact low-intensity ultrasonic technique to monitor methanol quality during production in real time. And I've shown you that for the compositions and for parameters we studied, this technique was able to detect changes in moisture content of the methanol in a wide moisture content range. And this technique was also able to precisely measure the methanol thickness, and we were able to measure even a fraction of a millimeter. We found a very strong correlation between the peak cutting force obtained from texture, which is an offline measurement, and ultrasonic velocity, which is online, meaning that we may not have to perform some of these offline measurements, and we can get all the information we need in an online manner. We also talked about a mechanical parameter that can be obtained from ultrasonic measurements, the longitudinal modulus. That can be directly measured using ultrasonic velocity, which I showed two different methods to measure it. If we know the methanol density, which can be measured independently very easily. And we also saw that there's some inverse relationship between ultrasonic velocity and protein quality indices, but we'd like to look more into whether we can find different relationships as we change the protein composition of our raw material. So that's a future project. So in the future, we would like to look at whether we can detect changes in quality at earlier stages of extrusion. So with the air-coupled transducers, we're very close to the die end, but we're looking at the end product. Sanitary and online, but it is still the end product. So the next thing we're going to be doing is looking at the use of contact transducers and see what is really changing in that long cooling down in terms of protein structure and that fiber and layer formation. We also would like to look at how we can manage the microstructure and texture in other ways when compared to just changing extrusion conditions. And for that, we're already looking at gas injection assisted extrusion which is injection of different gases like nitrogen, carbon dioxide, or air into the extruder barrel, so that these gas bubbles will stay as separate entities as the melt is flowing inside that long cooling die and add to the microstructure. And without changes in microstructure, some changes in sensory properties, textural properties are expected. For example, on this slide, we just looked at the 3D microstructure using X-ray microtomography. Just looking at the text center slice, and you can see that there's several bubbles that are oriented in the flow direction. And the question is, how that gas cell distribution will affect total porosity, and how the orientation of these bubbles will affect the sensory attributes of meat and logs. And finally, we're going to look at different feed mixtures and different extrusion conditions to strengthen the understanding as to whether ultrasonic measurements can be predictive of changes in protein quality in real time during production. And with that, I'd like to acknowledge my collaborators and my team members who's been working on this project. And I'd like to thank the Good Food Institute again for funding this project, and I'm happy to answer your questions if you have any. Thank you.
Wow, thank you so very much, Dr. Koksel, for an incredible presentation. The chat is absolutely exploding with gratitude for you taking the time to share these incredible findings with everybody, and also just for your seminar presentation style. So deeply informative and such a valuable use of an hour. Thank you so much, Filiz. So we do have lots of questions flowing in from the seminar attendees. Those of you who have not yet gotten the chance to ask your questions, please do feel free to drop those into the Q&A section of your Zoom bar, and we will try our best to get to them. The first question that I want to ask you is about maybe other non-destructive high throughput analytical methods that you know of that you could see having applications in alt protein or alt meat research. So could you couple any of these other methods with the ultrasound technique to assess even more product characteristics at one time?
Mm-hmm. I think I was in a conference last year in Singapore, and there's a great group from Europe. I cannot remember where exactly. I'll need to look up my notes, but they're using all sorts of waves from the electromagnetic spectrum, mainly on the infrared region, to look at properties of the plant-based meat alternatives during extrusion. I've been following and looking to see if they're published yet, but I don't think so. So I think soon there's a very strong group in Europe that I know of, but that's quite different because those are waves from the electromagnetic spectrum, whereas ultrasound is mechanical waves, so pressure waves, so very, very different techniques. But I'm hoping that we'll see more and more techniques using the electromagnetic spectrum. I know NIR, for example, has been heavily used for things like measuring the protein content, but just going a little bit higher or lower in that electromagnetic spectrum, you can detect different things, things in addition to protein content, and maybe things related to protein structure, which would bring in lots of insights into what's happening during extrusion of and protein texturization processing.
Absolutely. Thank you. That is a great segue into my next question around other types of food processing methods aside from extrusion. Do you think this ultrasound technique would also work well with some of these other manufacturing technologies like melting or wet spinning technology? And if so, what are some of the considerations involved?
Um, I think I'll need to see the equipment a little bit better to see if it really applies to it. But so what's happening with this ultrasonic technique is, so you have a transducer, you can have some air in between, and then your material, and then some more air, and another transducer. In between these two, you can have other layers of plastic or other materials, but it needs to be stable. And during that measurements, you're not supposed to have, like a hand coming in and going out. Like, you do not want that. So preparing this presentation, and I was actually hoping to find some collaborators here for application of the technique to cultivated meat, for example, to see getting that ultrasonic signal at different times during that process to see when it is ready, so that you do not need to keep testing to make sure that it is at its optimal. So that's one of the areas that I see this technology, being expanded to. I also thought about the shear cell. I mean, I think that the technique could be also applied to. So I have the shear cell, one side, one transducer, maybe on the other side, another transducer. Really will depend on how much of that sound wave you'll lose due to scattering and absorption, but these can all be tried. So this is just, I think, the beginning, and the technique is very well established in different areas of food processing. So it's just a matter of applying the knowledge to a different technique, and I'm happy to collaborate on that, on these areas if there's any interest.
Awesome. You've heard that, folks. If there are any cultivated meat researchers there in the audience, or folks working on shear cell technology and other processing methods, that you might be curious whether there's an application of these ultrasonic measurement techniques to your research, you know who to reach out to. That's an
I'll think more about wet spinning. So I'll make a note of that, and maybe that could be. I just need to see the instruments a little bit in more detail to make a comment, but I'm writing it down.
Awesome. There's another question here around the extensibility of this velocity data to other textural properties of meat analogs like hardness, chewiness, gumminess, etc. Can you kind of speak to how velocity data might be correlated or related to those other properties?
Yes, so I definitely see some important information that's going to come out, for example, in chewiness. So anything that's a mechanical property, I think we'll be able to link it with ultrasonic properties, because that's also, the ultrasound is measuring the mechanical properties at the end of the day. So the reason why I focused on what I focused on today was because we looked at a range of different moisture content. So, and I think it's a little bit difficult to make, bigger statements here, but more data is on its way, where we're looking at different raw materials, different screw speeds. All of that will play into the sensory profile of that meat analog, and we are working to link those with ultrasonic properties. So it's coming.
Can't wait to see the additional research that is underway. There's another question around whether you've considered applying pattern-based analysis or artificial intelligence to your data eventually to kind of expedite end product research and development even more.
And this is a great question, and artificial intelligence especially just keeps I see it everywhere, and unfortunately, it's not an area of I'm not an experienced researcher in that area, but definitely it's an area that I would like to expand on. So if I do not see who is asking the questions, but if that person has experience and is interested in the technology that I went over today, I'm very happy to connect and look for ways to make the process more streamlined and get those answers faster. So I'm happy to connect.
Fantastic. Thank you, Filiz. Here is a more granular question. So maybe 5 slides from the end of your deck. There's a question from the attendee about interpreting the intersection points of like V and N and how those can be interpreted. So what do you think those intersection points say about the meat analog? Are they an abstract parameter mainly used to compare different testing conditions, or what meaningful insights do you interpret from those intersection points?
So to extract the ultrasonic velocity, one of the missing things in those 2 different methods is the thickness of the extrudate. So you can simply measure the thickness of the extrudate by cutting a piece and then measuring it, using a caliper. But high moisture meat analogs tend to be really soft. So we've tried that too. And we found that we're really, squashing the sample a little bit while we're trying to get that measurement. And also, it was defeating the purpose of keeping our hands off the whole process. So our idea was we'll simply measure the thickness and use one of the methods to get the velocity. But then having, both of those methods giving you the velocity and both of them having the thickness inside, it just becomes a mathematical problem of like solving those equations without the need to measure the thickness offline. Or you can simply add on, a high-speed camera at the end of your extruder, and then, take images, and then you'll know the thickness. But again, that's another, I don't know, $25,000, $50,000 more analysis to extract that information. So it was really about what is it we can get from those 2 methods to obtain velocity and thickness without adding more equipment to the system. You like in the food industry, not all extruders and all processors will have the capacity and the funds to add a high-speed camera, right? So just using ultrasonics, you can get the information on thickness and the velocity. So basically those 2 curves and intersection is saying is from one of the methods, you get a curve as a function of different thicknesses, you get like different velocities. And from the other method, again, different as a function of different extrudate thicknesses, you get different velocities. And at that intersection point is that accurate thickness and the velocity of the sound in the meat analog. And while that's great to have that information on the velocity, which we later linked to mechanical properties, what we thought was a really nice discovery in this whole process was that with this ultrasonic technique in a sanitary manner without touching your extrudate, you can detect changes in product thickness, and I'm talking about fractions of a millimeter here, so it's very sensitive in real time. So you can think that maybe the meat analog thickness, is not something that will become very important for selling your product, but it tells a lot about consistency. So when you're changing some of your extrusion conditions on the go, by just looking at the changes in thickness, you can say, are things consistent? And that is very important for the food industry. To produce a high quality product is something, but to produce it the same way consistently is something completely different. And that is really important. And I think with this technique, you're able to do that with no problems. And we thought it was really interesting.
Absolutely. Yeah, it is fascinating. And I see a very strong value proposition. I mean, the batch to batch variation and so many of the inputs that go into the plant-based meat or alt meat production process, something we definitely want to control for. So appreciate that response. Thank you for those reflections. And kind of back to a former question around other non-destructive methods for studying the properties of meat alternatives. Another attendee has pitched maybe near infrared technologies or hyperspectral imaging technology, and is curious if you have any thoughts on those. If not, totally fine. But those are two other technologies that folks are throwing in the mix.
Are these techniques that I've tried? No. But certainly I can see some application. So the trick will be to make them real time. So again, not my area of expertise, but for example, for hyperspectral imaging, the question is going to be, you collect all that data, and will you be able to analyze it on the go to, tell you, okay, so this is not good, this is too soft, or this has low digestibility and whatnot. So I think like this work took 3+ years for us. So I think other techniques can obviously be used, and the more the better. But someone with an expertise in hyperspectral imaging will have to, spend some time to build that so that we will be able to use it. And given that different techniques give different information, why not, use both and get a better perspective of what's going on.
Absolutely. Thank you so much, Dr. Koksel. There is just a couple of questions left here. One is a quick one. An attendee is wondering whether air-coupled transducers are commercially available or only custom built.
The ones that we use are from a Canadian company. You could just simply put an order tomorrow and get them. But I'm not 100% sure about other places of the world, but I am sure there must be other companies. But if not, the Canadian one from Ontario, VN Instruments, they sell all sorts of ultrasonic equipment and awesome. Air-coupled transducers.
Great. So commercially available, unsure of universal accessibility, but certainly available. That's awesome. Thank you so much for taking the time again to share your knowledge and wisdom and research with this community. Before we wrap up today, I would love to just ask you whether there are any last thoughts you would like to leave this global community of scientists with before we wrap up our seminar.
I'd like to just thank one more time to Good Food Institute. You really supported me with funding this research, but during the first years of my career, where when I first got this research grant, and throughout all those years, the support has been amazing. So I do thank you very much for that. On the research side of things, I know me and my colleagues at the University of Manitoba are always looking for new trainees in the protein sector. We have a very good group working on food proteins from nutritional quality to processing and packaging and material science. So if in this community, if there are trainees who are looking for grad student positions or postdoctoral positions, please, especially in the, we're interested in alternative proteins sector, please reach out to me.
Wow, I'm sure we'll have lots of folks knocking on your door. And if we can kind of be helpful in advertising those open positions more broadly, feel free to let us know. We have an industry careers board that we're happy to post those on. Thank you again so much for your time. Everyone, before I let you all go, I did want to shout out a few quick announcements for those of you that are hungry for more learning. The first is that we have a GF Ideas plant-based meat networking mixer happening on March 30th. So if you're an entrepreneur, a scientist, investor, policymaker, or innovator interested in the plant-based meat industry, like I assume all of you here are, do join us on March 30th for a networking event where you can share ideas, keep this discussion going, and make new connections with others in this very rapidly evolving field. And the other is that GFI APAC just unleashed a new report called The Untapped Potential of Mung Beans for Alternative Proteins, which is a super comprehensive exploration of the enormous opportunities and solvable challenges in utilizing mung beans for alternative proteins. So highly recommend you all diving into that report as well. And then next month, we will again have our monthly Science of Alt Protein seminar, this time taking a deep dive into the environmental impact of cultivated meat production, where CE Delft and GFI's Elliot Swartz will be talking about their updated cultivated meat life cycle assessment that was recently published in the International Journal of LCA. So be sure to join us on April 12th for that discussion as well. Thank you all again so much for attending today. Massive thank you to Dr. Filiz Koksel for allowing us to be a part of your research and elevating the tremendous progress you're driving forward for alternative protein science with our community. And I hope you all have a fantastic rest of your week. Thanks so much, everybody. Bye.
Thank you very much, Amy.